Scientists have constructed the most complete and detailed single-cell map of embryo development in any animal to date, using the fruit fly as a model organism. Published in Science, this study, co-led by Eileen Furlong at EMBL and Jay Shandur at the University of Washington, harnesses data from over 1 million embryonic cells spanning all stages of embryo development and represents a significant advance at multiple levels. This fundamental research also aids scientists' ability to pursue questions like how mutations lead to different developmental defects. In addition, it provides a path to understand the vast non-coding part of our genome that contains most disease-associated mutations. Just capturing the entirety of embryogenesis, all stages and all cell types, to obtain a more complete view of the cell states and molecular changes that accompany development is a feat in its own right, said Eileen Furlong, head of EMBL's Genome Biology Unit. But what I'm really excited about is the use of deep learning to obtain a continuous view of the molecular changes driving embryonic development, down to the minute. Embryonic development begins with the fertilization of an egg, followed by a series of cell divisions and decisions that give rise. Researchers have been studying this process of embryonic development for over 100 years. But only in the last decade have new technologies enabled scientists to identify molecular changes that accompany cell transitions at a single cell level. However, attempts to profile the entirety of embryo development at single cell resolution have been out of reach due to many technical challenges in sampling, costs, and technologies. In this regard, the fruit fly, Drosophila melanogaster, a preeminent model organism in developmental biology, gene regulation, and chromatin biology, has some key advantages when it comes to developing new approaches to address this. This, in combination with the many discoveries made in fruit flies that have propelled understanding of how genes and their products work, encouraged the Furlong Lab and their collaborators to take on this challenge. Our goal was to obtain a continuous view of all stages of embryogenesis, to capture all of the dynamics and changes as an embryo develops, not just at the level of RNA but also the control elements that regulate this process, said co-author Stefano Secchia, a PhD student in the Furlong Group. Preliminary work with enhancers in 2018. The Furlong and Shandur groups showed the feasibility of profiling open chromatin at single-cell resolution in embryos and how these DNA regions often represent active developmental enhancers. Such a map is essential to understand what drives specific aspects of embryonic development. I got really excited when I saw those results, Furlong said. To go beyond RNA to look upstream at these regulatory switches in single cells was something I didn't think would be possible for a long time. Going beyond snapshots, the 2018 study was state-of-the-art at the time, profiling approximately 20,000 cells in 3 different windows of embryo development, at th. The team therefore explored the potential of using samples from overlapping time windows, and as a proof of principle, applied the concept to one specific lineage, the muscle. This then set the stage to scale up dramatically using new technology developed in the Shandur lab. The team's current work profiled open chromatin from almost 1 million cells and RNA from 500,000 cells from overlapping time points that span the entirety of fruit fly embryo development. Using a type of machine learning, the researchers took advantage of the overlapping time points to predict time at a much finer resolution. This could pinpoint exactly when, and in which cell type, a mutant's phenotype arises, as the researchers showed in the muscle. In other words, this work not only helps to understand how development normally occurs but also opens the door to understanding how different mutations can mess it up. For example, mammalian embryo development, in vitro cell differentiation, or even post-drug treatment in diseased cells, where gaps in sampling times can be designed to facilitate optimal time prediction at a finder resolution. Going forward, the team plans to explore the atlas's predictive powers. Combining all the new tools at our disposal in single-cell genomics, computation, and genetic engineering, I would love to see if we could predict what happens to individual cell fates in vivo following a genetic mutation, Furlong said. But we're not there yet. However, before this project, I also thought the current work wouldn't be possible anytime soon. Diego Calderón, Ronnie Blecher-Gonen, Xingfan Huang, Stefano Secchia, James Kentro, Riza M. Daza, Beth Martin, Alessandro Dulja, Christoph Schaub, Cole Trapnell, Erica Larson, Kate M. O'Connor-Giles. The Continuum of Drosophila Embryonic Development at Single-Cell Resolution. Science, 2022. 377, 6606, doi. 10. 1126, Science. ABN 5800 European Molecular Biology Laboratory. European Molecular Biology Laboratory. 2022, August 4. Single-Cell Approaches and Deep Learning to Map All Stages of Fruit Fly Embryo Development. Science Daily. Retrieved August 5, 2022 from ift.tt/3ddSTLC European Molecular Biology Laboratory. Single-Cell Approaches and Deep Learning to Map All Stages of Fruit Fly Embryo Development. Science Daily. https://ift.tt/3ddSTLC, accessed August 5, 2022. Thanks for watching. Please subscribe my channel for more videos.